Thank you for the introduction. My name is Ted Hong Chan-young in Korean. And first of all, I'd like to thank Brick and Hanbisa for the opportunity to share my research today, and thank all of you for joining my talk. And today's title is going to be a little bit different than the announced, but it includes, it is just a little broader. It is going to be the multi-omics investigation of the alteration of human gene regulatory networks by human virus transcriptional regulators. And I will present in English, and I think we can have a Q&A in Korean. And before I present the story, I have to clarify that this work that I present today is done in Cincinnati Children's Hospital Medical Center, my former affiliation. And it is not related with my current affiliation at all, the AstraZeneca. So please note that I cannot mention anything about my current affiliation work right now. And I want to start with some background information. As many of us know, the crosstalk between the genetic risk polymorphisms and environmental factors is thought to influence the onset and the progression of many human diseases, such as cancers, cardiovascular diseases, and multiple sclerosis or systemic lupus like autoimmune diseases. However, the detailed mechanism of this interaction remains largely elusive for most of the diseases. Virus infection are a known environmental factor that are closely linked for many human diseases. For example, obviously, the SARS coronavirus 2, which caused the COVID-19, and HIV with AIDS, human papillomavirus with cervical cancer and head and neck cancer, and Epstein-Barr virus, EBV, with Burkitt's lymphoma and Hodgkin lymphoma, or the other cancers. Over the past 15 years, a multitude of genome-wide association studies, GWAS, have identified more than 15,000 disease-associated genetic variants for many disorders. However, 90% of these variants fall within non-coding region of the genome, which implicates a key role for regulatory proteins such as transcription factors in etiology of many human diseases. And one way host-virus interaction can occur through the alteration of the transcriptional regulation by virus-encoded regulatory protein. I want to call it as a VTRs. And those VTRs can regulate gene expression of many human genes, including immunosuppressed genes such as TGF-beta activation by EBV protein ZTA, or pro-inflammatory genes such as induced lung inflammation by N protein from SARS coronavirus 1, the original SARS, and activation of the cell cycle and the proliferation genes by E1A from adenovirus, and many other oncogenes and tumor suppressor gene regulation, including the MYC regulation by adenovirus, EBV, HPV, and other herpes virus, and TERT gene regulation by EBV and HPV and other many viruses. And these VTRs sometimes can directly bind to the nucleic acid, and for example, as the HPV-E2 protein, which can bind to the promoter region of the human IL-10 and induce its expression. Or sometimes these VTRs requires the human DNA binding factor from human protein, because it has transactivation domain, but not the DNA binding domain. And VTRs can hijack the transcriptional regulation machinery from human side, which is TET. What does TET do in the HIV infection, and also modification of the chromatin regulation by these proteins. And in previous study, we have identified the 419 VTRs that are encoded by virus, which can infect humans. And here's a plot from that study, which shows the number of VTR per each viral families. And as we can see, there is a SARS coronavirus here, which has the N protein and E VTR protein. And there is EBV, which has the most VTR in its genome, which will be today's topic. So Epstein-Barr virus is DNA, double-stranded DNA virus, which is ubiquitin surface virus, which means almost every adult population has the EBV infection. And previous studies showed the roles for EBV in mononucleosis and several hematological cancers, including Burkitt's lymphoma, Hodgkin lymphoma, and other types of cancer as well. EBV is also the most frequently reported environmental factor for autoimmune diseases. For example, in multiple sclerosis, EBV infection is greater in patient than healthy individual. And the fresh new study, which was published in Science last month, also showed that the link between EBV and multiple sclerosis. And in systemic lupus, EBV is known to increase the disease risk as much as 50-fold. However, despite those evidences, the understanding of the molecular mechanism of how EBV host interaction increase the autoimmune risk is still limited. EBV can encode several virus transcriptional regulator, including ZTA, Epstein-Barr virus nuclear antigen 1, MNET1, MNET2, MNET3, and RTA, and other protein as well. For example, as I mentioned, there is a direct binding from the VTR, and in this case, the ZTA from EBV can directly bind to the human genome and induce its expression. And MNET2 from EBV can bind to the other DNA binding factor from human and then induce human gene transcription. As I previously mentioned, MNET2 is known to regulate human B cell and epithelial cell gene transcription. And it is known to be involved in immortalization of EBV-infected B cells. And it requires binding partners such as RBPJ, SPY1, EBF1, all human transcription factor. In detail, MNET2 can mimic the intracellular domain of Notch1. When the intracellular domain of Notch1, which is called NICD, enter to the human nucleus, it can bind to the RBPJ and activate the silence form of the RBPJ and induce its target gene expression. And similarly, MNET2 can bind to the RBPJ and it can induce the gene expression of the human gene, which is the same target as NICD, including the viral oncogene LMP1, but also many human genes, including CMYC, HAS1, and BCL2 type of genes. And Notch signaling, these Notch signaling and RBPJ are known to play a key role in the onset and progression of autoimmune diseases, including multiple sclerosis and lupus, which suggests maybe MNET2 can play the similar role. A previous study from my PhD lab revealed that a significant number of autoimmune-associated genetic variants are located within MNET2 ChIP-seq peak. In detail, in order to get these integrative information from ChIP-seq binding information, and also the genetic information from the GWAS study, the lab created an algorithm called regulatory element locus intersection, which is the permutation program. Using the 1544 human protein ChIP-seq dataset from the GEO gene expression omnibus database, and disease-associated genetic variant information from the GWAS from more than 200 diseases. RELI, the permutation algorithm, works as following. First, we expanded the SNPs by LD expansion. And then we randomized the input. in bed file in each iteration for the permutation test. And in every iteration, we calculate the overlap between the TF and the input. And we do the n times of iteration. Sometimes we do 500, but in simulation, 2,000 times is the best. So we settled down as a 2,000 times until we have estimated intersection of the significance, where we can calculate the p-value by calculating the mean of all overlap to the original overlap, which we can get the z-score that can be converted into p-value. So here's an example of the ReLI output. In here, blue means there are one or more intersection in a locus, which is x-axis, and with the TF. And strikingly, here's the output for systemic lupus. And strikingly, the previous work could find near half of the SLE risk loci are occupied by the EBV protein, MNET2, which is pretty consistent evidence that MNET2 or the EBV can play a role in autoimmune diseases. Not only the lupus, but also in multiple sclerosis, MNET2 showed 44 overlap out of 109 MS-associated genetic variants, with almost 5 times enrichment with extremely low p-value. So the understanding the interaction between MNET2 and human genome is important to comprehend the development and progression of autoimmune diseases. Then what is the underlying molecular mechanism for MNET2 host genome interaction? On the other side, EBF1, which I previously mentioned, is a human transcription factor, which is known to opening the chromatin, so it is called pioneering factor. And MNET2 RBPJ complex can recruit the EBF1, which leads to the hypothesis that MNET2, along with the RBPJ, might act as pioneering factor or the cofactor. Also, it had been shown that MNET2 can form new chromatin looping interaction near the MYC promoter, which supports the role for MNET2 in chromatin looping interaction. However, despite those findings, a genome-wide investigation of MNET2-dependent differential chromatin modification has not been reported. Now, in this project, we explore the roles of MNET2 within the human B-cell gene regulatory network by examining MNET2-dependent alteration to human chromatin landscape and investigating the impact of disease-associated genetic polymorphism on each of these mechanisms. And the previous Nature Genetics work was using to ChIP-seq data. And in this project, we use Ramus B cell that are EBV negative B cell, which can be infected with EBV. And we used both of MNET2 positive EBV strain called B958 and MNET2 negative strain called P3HR1, which can be infected to Ramus. So we can, not only we can just recapitulate the previous work, but we can control the EBV infection and also the presence of MNET2 protein. So again, overall experimental design is like this. We have uninfected, MNET2 negative EBV infected, MNET2 positive EBV infected. We conducted RNA-seq, ChIP-seq to answer whether MNET2 can really express, really induce its gene expression in the B cell, and also ATAC to answer the question about the chromatin modification and the looping structure by performing high ChIP-seq. First, we performed the RNA-seq with the uninfected and MNET2 negative EBV and MNET2 positive EBV. We used this program called STAR to align the read, and counted the read with the feature count and calculated the differential gene expression using DESeq2, the popular tool. And I have to acknowledge that wet lab work was done by Daniel Miller for RNA-seq. And differential gene expression in EBV MNET2 positive EBV infected compared to the uninfected looks like this, which show the same gene as the previous reported, but also many other genes, many other noble genes, which shows that our experiment possibly worked and was consistent. As a total, we had 493 genes that were differentially expressed in MNET2 positive versus uninfected. And likewise, we also calculated the differential gene expression comparing MNET2 negative versus uninfected, which was in total 171 genes. And among these upregulated genes and the downregulated genes, we wanted to focus the genes that are only differentially regulated in MNET2 positive set. So we compared these MNET2 positive side to the MNET2 negative side and then subtracted out these MNET2 negative side. So these 423 genes from the upregulated side and also 178 genes from the downregulated side. We designated those 421 genes as MNET2 DGs. And I want to call these as MNET2 DGs from now on. First, we looked at whether the MNET2 DGs showed a significant overlap between autoimmune SNPs. That was our one of the most interested questions. So we not only used the just gene body region or the exon region, but also we padded some searching window from the transcription start site, TSS, because sometimes enhancer can bind very far away, like 200 kb. So we could include all interaction. And then we ran rely to the permutation test. So here is an output for the first rely that we can see today. X-axis shows the disease intersected with the MNET2 DG, and y-axis is corrected p-value, negative log 10 corrected p-value for the 171 diseases that we tested. And here's a all autoimmune disease SNP, which was significant than random. And many other autoimmune diseases like systemic lupus, multiple sclerosis, and previously defined MNET2-associated disorder by the Nature Genetics paper. And also there's a Hodgkin lymphoma, which is known to be related with the ABV infection. So it seems like our work, our RNA-seq worked. And here's a table view, which is just the same as our previous bar graph. But I want to mention that the overlap diseases are the same disease that we found in the previous work, the Nature Genetics work, but using very different data set and the approach, not public ChIP-seq data set, but controlled in-house generated RNA-seq data set. And we also compared the intersection between our MNET2 DG, padded MNET2 DG, with the public ChIP-seq data set that we used for the Nature Genetics paper. And it showed, it turned out that it is very highly, significantly overlapped with the other MNET2 ChIP-seq, which is reassuring that our work is done well. So we conducted, but we wanted to know whether MNET2 can actually bind and regulate the human gene expression in Ramus cell by conducting the ChIP-seq. So we conducted the ChIP-seq. We compared to the MNET2 antibody to the input. We aligned the read using bow tie and called the peak with using MAX2, and a program called Manon was used for differential peak analysis. And in total, we had the 4,219 peaks that were called. And we also wanted to know whether these MNET2 DGs were significantly intersected with the Ramus ChIP-seq the new ChIP-seq. So we did the same thing, running Rely, the permutation test. And the padded region of the 421 MNET2DGs showed significant overlap with the MNET2 ChIP-seq pig. And the very stringent intersection, which is 5 kb, the promoter intersection, showed the significant amount of enrichment, and also distal intersection, which is the 100 kb padded, which means our RNA-seq is highly intersected with the ChIP-seq pig. And we wanted to know whether Ramos ChIP-seq pig, how does it do with the other human transcription factors, as we looked at in the Nature Genetics paper. And the similar results were found. Significant overlap were found with the other human transcription factor, including the known MNET cofactors like RBPJ or NF-kappa-B, relay. And other chromatin pioneering factors like EBF1 and SPY1 with the extremely low p-value. Computer couldn't, sometimes computer cannot calculate some numbers which is super low or super high, and it exceeded that limit. So EBF1 and SPY1 was found to be very significant, and also known factors for the chromatin looping formation like YY1, RED21, and CTCF in our ChIP-seq pigs. These nomination of chromatin-related factors intersected in our permutation test led the hypothesis that MNET2-driven alteration of human chromatin landscape. So we conducted the ATAC-seq and high ChIP-seq to get the effect of MNET2 in chromatin regulation. We used the same condition as RNA-seq and called the pig and calculated the differential pig using very similar pipeline as the ChIP-seq analysis. And in ATAC-seq, we could get the 1547 and 690 MNET2-dependent open and closed chromatin region, respectively. And these plot shows that there is huge pileup in MNET2 positive, where uninfected MNET2 negative showed just medium of the pileup in positive side, which is open. And also the negative side, MNET2 showed quite a lot of less amount of peak compared to the others. And we designated those differential region as a MNET2-dependent open and closed chromatin region. And here is a genome browser view of MNET2-dependent open chromatin region, where MNET2 positive ATAC, which is red one, showed peaks, but not much in uninfected, the gray, or the negative, MNET2 negative, which is blue. Oh, sorry. And which has the clear ChIP-seq peaks from all cell types, Ramus, IB4, and public GM12878, and also in-house generated ChIP-seq peak for GM12878 that are located in the promoter region of the most upregulated MNET2DG, which is this non-coding RNA, which is quite promising. MNET2 binds, opens, and upregulates the genes. Using Homer, we wanted to see what kind of motif, transcription factor motif, were enriched. And we not only just used the default Homer motif, but my former professor was the inventor of the CISBP, which is most used the transcription factor motif database. And we had we have so many motifs from the same transcription factor. And in our MNET2-dependent open region, from the ranking 1 to ranking 20 was just CTCF, CTCF, and CTCF. And also RED20 was also super significant. Similarly, but even robust finding was found in the real ChIP-seq data. When we ran the Rely, we could see the CTCF, RED21, SMC3, YY1, which are known to modify the chromatin looping structure, which reassures and highlight the possible roles for MNET2 in modifying chromatin looping structures. So in collaboration with the Dr. Bo Zhao's group in Harvard University, we conducted the HiChIP-seq and we analyzed the data. Pipeline was a little slightly different. We called the HiC using HiC Pro, and then we integrated with the HiC with the H3 K27 acetylation chip-seq peak, which is used for the designation of the regulatory region using HiChipper. And then we calculated the differential loop using deep loop program. And as a result, we could calculate the 1433 and 311 energy-dependent gain and loss of chromatin looping, which can be shown in here. And for example, we can see that MNAT2 binding signal here within a gene body region of one gene called SLAMP1, which loop the promoter region of that gene and regulate gene expression. We could see that differential gene expression. This is also one of the most strongly upregulated genes from MNAT2-DG. And here's the same view, but very zoomed out version. This is gene body and the promoter region. And remarkably, nearby the SLAMP1 locus, it contains a lot of robust MNAT2-dependent looping events involving multiple genes, revealing extensive MNAT2-dependent rewiring of chromatin three-dimensional landscape at this locus. And this is just only an example of the finding. We could see a lot of extensive rewiring of chromatin looping event by the virus transcription factor to the human genome. So our data suggests three mechanisms of MNAT2-dependent gene regulation, which is bind to the promoter enhancer with the cofactor, or alter the chromatin accessibility, or form the novel chromatin 3D interaction. But how about the disease mechanism? So we wanted to know the impact of disease-associated genetic variants. So we ran the permutation test with the entire SNP to each of data set that we had: RNA-seq, chip-seq binding information, chromatin accessibility, and the chromatin looping. MNAT2-DG, mostly upregulated side, but also some downregulated side, showing of multiple autoimmune-associated SNPs. The red shows the significance, and the upper number are full change of the enrichment, and the lower number is actual overlap. And we can see that the upregulated side tend to have more significance with the disease-associated genetic variants. If we combine these, this is the same plot, the same bar plot that we saw about 15 minutes ago. And here is a plot for MNAT2-dependent gain of looping and also the loss of looping compared to the SNP. And these looping interactions shows that gain tends to have more enrichment within the autoimmune-associated SNPs from many different autoimmune diseases. However, we couldn't see any significant intersection except a multiple sclerosis of the opening side of chromatin accessibility, which was very shocking to us because our other data showed that upregulation or the gain of loop showed the significant intersection, while the open chromatin didn't show almost at all. So these results prompted us to further examine the relationship between chromatin accessibility and autoimmune risk loci. So we investigated the relationship between autoimmune SNPs and ataxic peak, not only from MNAT2-dependent set, but all infection type, which means we individually looked at uninfected chromatin accessibility, and MNET2 negative accessibility, and MNET2 positive accessibility, and we calculate the significance of the intersection between the SNPs. And it turned out that it was even significant in here, there, and also MNET2 positive side. So we couldn't get the differential effect because all SNPs were significantly intersected within all open chromatin region, which means autoimmune risk variant tend to be located in the chromatin region that are already open regardless of EBV infection type, no matter what the infection type, so like uninfected or MNET2 positive. And these analyses revealed the significant intersection between autoimmune-associated genetic variants and region of the genome that are constitutively open in B cells, which was very opposite finding that we previously thought, but very novel finding. So collectively, these data indicate that MNET2-dependent de novo alteration of long range of chromatin interactions is highly associated with autoimmune disease-associated genetic variants, which reveals the key role for MNET2-altered chromatin interactions in autoimmune disease etiology. On the other side, previous analysis revealed that MNET2-dependent binding can be significantly enriched for autoimmune risk loci by Allylic manner, which means it can be one allele can show this amount of peak, while the other allele, the heterozygous allele, show more or less binding. So previously, our lab also developed a pipeline called Measurement of Allylic Ratio Informatics Operator, MARIO, by Dr. Mario Pujato, to determine the allylic binding locally with a given variant, which weighs the imbalance between the number of the reads mapped for each allele in heterozygous. For example, let's say this is 30 and this is 10, then the ratio or the imbalance is 3:1. And the total number of reads mapped at this reads variant loci, which is 40 in this case. And also the number of consistency of available experimental replicates. I think the internet was not too good. Sorry for that. And in this study, we identified the 18 new instances other than the Nature Genetics paper of the allele-dependent MNET2 binding and autoimmune risk loci. For example, the ZMIS1 gene called ZMIS1, its expression were threefold lower in MNET2 positive side compared to the uninfected, and even the MNET2 negative set showed even higher expression. So it was totally downregulated by the MNET2 protein. And MNET2 ChIP-seq peak showed that the allylic binding to the this RS1250567, which was MS-associated genetic variants, which we could see that it tend to have more reads on the T side, not the C side, which is located in the gene body of ZMIS1 that loop to the promoter of each isoform, so shorter form or the longer form. And I couldn't capture, but the longer interaction is here to the other very, very long isoform of the ZMIS1. And this RS1250567 is also a strong EQTL for ZMIS1, which means it can be allylically expressed in EBV immortalized lymphoblast and also the full blood, which suggests the hypothesis that MNET2-dependent chromatin alteration can induce these allele-dependent binding or the expression. In summary, we nominated the possible molecular mechanism of MNET2-dependent regulation of human gene regulatory networks, which are binding to the promoter or enhancer, which is already known a little bit, but also to alter the chromatin accessibility genome-widely, and also forming novel chromatin 3D interaction. We also demonstrated that many of these mechanisms significantly intersect with autoimmune-associated SNPs and identified the multiple EBNA2 interaction, which are allele dependent. It is possible that other viral transcriptional regulator play similar roles in other diseases. As I mentioned, as in previous slide, we have identified 419 VTRs only in human virus. And the recent study, this is also from last month, right? Last month. Recent studies show that there are 10 to the 5 novel viruses, even just in RNA virus species. So there should be a lot of novel VTRs as well. And preprint study found that chromatin looping alteration by COVID SARS coronavirus 2 doesn't know whether the VTR is involved in here or not, but virus can alter the human chromatin architecture supported by the other virus as well. Maybe by VTR, who knows? So future studies will deepen our knowledge of mechanism underlying virus-host interaction, and even further to the other host microbial interactions, like maybe plant fungus or microbiome, I don't know. And ultimately, they will provide both a rationale and foundation for therapeutic approaches targeting these interactions. So I want to thank all the lab members, especially my PI, my former PI, Dr. Matthew Wyrak, and also the Dr. Srija Parameswaran, who did a lot of work after I graduated, and also the lab technician who did the hard work for me, and the collaborators as well. And I want to thank all of you for your time, and thank you for your attention.